This is a Professor CBMD PhD, and today's Saturday, May 17th, 2025. It is 5:36 and 38 seconds, a.m. Eastern Standard Time, and today's research, histones. So let's hop to it here. Chromatone remodeling is a term used to describe the process of displacing histones to control access to DNA. In some cases, this means that cells simply slide nucleosomes from one position in chromatome to another, which can change the spacing between nucleosomes. In other cases, entire nucleosomes are temporarily removed from a particular region of DNA. Large ATP adenosine triphosphate consuming complexes are primarily responsible for performing these remodeling activities. Humans have at least 8 different complexes, and the best known complex is called SWI-SNF. The beads on a string structure we see in a microscope only appears when we break cells open and spread out their contents. In intact cells, chromatosomes are clustered together in a highly ordered fashion to form a series of similar configurations, all of which are called the 30 nanometer fiber. In prokaryotes, similar 40 nanometer fibers form from clusters of the beads in the genophore. These fibers represent level 3 of DNA organization. Multiple configurations have the same name because scientists are still not entirely sure how many of the configurations are present in living cells. The electron microscope, which is a common tool for observing chromatin, cannot be used to view living cells. These fibers will form spontaneously in the test tube if the salt concentration of the buffer is kept low enough, demonstrating that no additional proteins or metabolic energy are required. In simpler terms, a chromatosome is a nucleosome attached to a linker histone. The important distinction to keep in mind is that all of the mechanisms discussed here about controlling DNA access focus on the shape of the nucleosome, not the chromosome. This means that loosening of DNA around the spool, sliding DNA over the surface of the spool, and even removing the spool altogether takes place when the linker histone is not present, and thus this is all happening to a nucleosome. After the modification has occurred, the linker histone returns to bind the nucleosome. It again becomes a chromatosome. DNA reforms a 30 nanometer fiber when chromatosomes bind together via their linker histone. The 30 nanometer fiber is held together by electrostatic interactions between different histones. For example, a negatively charged region on histone H4 binds to a positive region at histone H2A H2B complex in another nucleosome, drawing the two nucleosomes together. And further compacting the chromosome, in addition, linker histones bind to each other, as well as to other proteins that serve as bridges between chromatosomes. This results in addition shortening and thickening of the chromosome and increases the packing density to about 42 times that of double-stranded DNA alone. At level 4 of DNA organization, the 30 nanometer fibers are attached to a protein RNA scaffold, also called a matrix, that keeps them organized. Specifically, the fibers are attached to the scaffold at intervals of 10 to 30 microns, thereby forming so-called loop domains of approximately 60 kilobases in length. Exactly how these loops are attached to the scaffold is not known, but the attachment occurs at DNA sequences called Mars or SARS for matrix or scaffold attachment regions, respectively. These sequences typically contain a large number of A-T base pairs, but are otherwise not very similar. Many proteins have been isolated from the chromosome scaffold, and the structure is sensitive to enzymes that digest an unknown form of RNA. But it is not yet clear how these molecules assemble to form the mature structure. Loop domains are approximately 750-fold more compact than bDNA. The prokaryotic chromosome is thought to consist entirely of level 1 to 4 structures. This video was created for educational purposes only. Codename Will. Professor Christopher Brunet, leader of research, scientist, ufologist, psychic, computer programmer, computer technician, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. Passion, world peace. Hope you enjoyed listening. Until next time, have an excellent day. This is a Professor C.